Hi, and welcome back. So in this video, I'm going to talk about a phenomenon called linkage. So linkage occurs when two genes are so close together on a chromosome that they get inherited almost as a single inheritance unit. So basically, when one gene goes, so does the other. And that is one of those things that sort of works against Mendel's law of independent assortment, right? That says that all genes are inherited independent of other genes, and that's not always the case. And linkage kind of disrupts that. Then we're going to also talk about the fact that we can actually use the fact that linkage exists to to analyze the architecture of a chromosome. Because if we know that, these two genes are linked and this one's only partially linked, we know where they are kind of relative to each other physically on the chromosome. So it's kind of a cool old school technique for mapping chromosomes that we did before we had the ability to sequence genomes and things like that. So let's talk about recombination first. So this is all about recombination. So there is interchromosomal recombination and there's intrachromosomal recombination. So when we talk about interchromosomal recombination, let's back up. Recombination, we're talking about the shuffling of genes. Okay, the shuffling of genes. So interchromosomal recombination happens during meiosis. And it happens during metaphase 1, where you have that independent assortment. So if you recall, in meiosis, during metaphase 1, you've got those tetrads lined up. So you've got two pairs of sister chromosomes that are homologous to each other. And then they kind of line up randomly so that like mom's chromosome 1 goes this way and dad's goes this way, but mom's chromosome 2 goes this way and dad's goes that way. And through that independent assortment, we get a recombination of genes so that you end up with a mixture of the parental genetic information being put into those daughter cells, which end up becoming sperm or egg through the process of meiosis. Okay. But there's also something called intrachromosomal recombination that happens. And this is going to happen during prophase 1 of meiosis. And this is what happens during crossing over. So if you recall, during prophase 1 of meiosis, you've got those pairs. So dad's sister chromatids of homologous chromosome 1, the mom's chromosome 1, and then these guys cross over, right? You end up getting those holiday junctions, and some of dad's chromosome ends up on mom's, and some of mom's ends up on dad. We kind of mash up the information that way. When we talk about linkage, we're going to be focusing on the latter. We're going to be talking about the intrachromosomal recombination. That's how we're going to follow this linkage process, because what we're going to learn is genes that are linked often cross over together, and that's why they end up getting inherited as the same type of a single inheritance unit. So while we'll spend most of our time talking about intrachromosomal recombination, let's talk just quickly about interchromosomal recombination so we can understand what we're talking about. So first and foremost, when we're talking about interchromosomal recombination, we are talking about the mixing of genes that are on separate non-homologous chromosomes, which means by definition, they are always unlinked. You cannot be linked and be on different chromosomes. And the genotype we get depends on the orientation of the chromosomes during metaphase 1. So let's look at this particular set of sex cells coming together. We get big A, little b, and then little a, big B. So these are what we would refer to as the parental genotypes. They then fuse together. This is the offspring. So this would be like the zygote for their children, their child, okay? And then at some point during that child's life, they're going to develop their own gametes, okay? And this is where we do that sort of meiosis 1 type thing and go, okay, what are the possible combinations that can come out of this mixture during metaphase 1? How can they randomly line up? And what happens during the process of crossing over? So there's some things that can happen, okay? First, we can get the parental genotypes back out, okay? So remember that what went into this was a big A, little b combination and a little a, big B combination. Well, just for sake of argument, dad, mom. Okay. Now the child is making their particular gametes. And if they get AB, big A, little b, or little a, big B back out in their sperm or egg, all they've done is thrown back out the parental genotype. They've thrown that back out. But if crossing over happens in the region where these genes exist, you could end up with a combination of, so here's that chromosome that used to have the little B on it, now it's got a big B because of crossing over. Here's that chromosome here that used to have the big B on it, now it's got a little b because of the crossing over event. These are what we call the recombinant genotypes. Okay? These are the recombinant genotypes. And the only way we can get a recombinant genotype is if crossing over occurs at that locus. So locus is a location on a gene. The plural of that is loci, which is a bit confusing, but that's what we do with Greek words that end in US. All right? So parental genotypes, recombinant genotypes. If you see a recombinant genotype, you know that a crossing over event has happened at that particular locus or those loci, depending on how many genes you're looking at. When we talk about linkage, okay, this is your expected probability, 50-50. So your expected recombination frequency of genes that are unlinked is that of an interchromosomal recombination, which is 0.5. Okay, so 0.5 is your baseline. You shouldn't be able to get higher than 0.5, although experimentally you may, because experimentally is always different than theoretical. Okay, or you can get lower. And as you get towards full linkage, you get to zero recombination frequency. So if you imagine, which we're going to in a minute, that A and B are right next to each other on the chromosome, well, if big A goes over to the other chromosome, then little b goes right with it. And if little a goes over to the next chromosome, big B's right behind it because they're right next to each other. It's hard to get a crossing over event that stops right after A but doesn't interrupt B. Okay? So when you have genes that are completely linked, you don't get these recombinant genotypes. But in some cases, you get an intermediate mixture. You get maybe a little bit more of parental and a little bit less of the recombinant than you expect, and that's what we call partial linkage. So let's look at that as it's happening, okay? So intrachromosomal crossing over, recombination, happens when genes are on homologous chromosomes. The word we use to describe that is syntenic. So if you hear that genes are syntenic, that means they are on the same chromosome as each other. It does not mean that they are linked, because if they are far enough away physically on that chromosome, they can't display linkage, okay? Recombination occurs during crossing over in prophase 1, right? So there's our, we'll just say dad's chromosome, mom's chromosome 1, crossing over happens. If crossing over happens, you end up with the recombinant genotypes, just like I described, big A, big B, little b. But the parental genotypes, if no crossing over occurs, you'll end up with AB and AB. Now notice the only difference between this and the figure I just showed you is, in this case, the genes are syntenic. They're on the same chromosome. We're not talking about two different chromosomes where crossing over can occur, okay? So what happens if we end up getting linkage? Well, we'll stop seeing these recombinant genotypes. Now, another thing that's always curious for us, because remember, right now, we have decided big A, little a, big B. But we might not know in a living organism the arrangement of those genes, okay? So this leads to a particular type of terminology that I want to make sure that we cover, and we'll talk about how we can learn more about that through this type of analysis in just a minute. If we end up with what we usually determine as the wild type allele, okay? In this case, just let it be the capital letters. Let it be the dominant alleles, okay? If both dominant alleles are together on one chromosome, and both non-wild type or recessive are together on the other chromosome, we call that the coupling configuration. So if you look over here, this is what coupling configuration looks like. Both little a and little b are together, big A, big B. Or in the case I just showed you here, this is what we call the repulsion configuration, big A, little a, big B. Now again, we know this because it's in the picture. In real life, we might not know that, and we might want to find that out, and we can actually use linkage analysis to try to figure this out in some cases. Okay, so recombination frequency. I've talked about it. Let's discuss exactly what it is. Recombination frequency is the number of recombinant gametes divided by the number of recombinant plus the parental. Okay, so just to back up, there are two different recombinants over 2 plus 2. So if you get 50-50 right here, you end up with a recombination frequency of 0.5 or 50%. Okay. If you see that, you can say that those genes are unlinked. Anytime you have genes that are non-syntenic, as in not on the same chromosome, they're going to have a recombination frequency of 0.5. Now, we use this to say approximately 0.5. Again, real numbers are never perfect. You might get 0.4888. You might get 0.5222 when you do your analyses. You know where you're at, all right? There's always a difference between theoretical and reality, okay? So if we get no recombination frequency, as in we don't see any of those recombinant gametes, those red letters go away and it's all blue, we just get the parental genotypes right back out, well, that's going to be complete linkage because you're going to get a recombination frequency of 0, okay? If you get somewhere in between, if you have a recombination frequency of 0.3 or less, trending downwards towards 0, okay, we call that partial linkage. That would indicate that those genes are syntenic, they're on the same chromosome. They are relatively close to each other, but they're not close enough to be completely linked. So crossing over events do happen in between those genes. The smaller that number gets, the smaller the distance between the two genes on the chromosome. So for example, if you're looking at multiple genes and you see one has a recombination frequency of like 0.1 to gene A, so B has a recombination frequency of 0.1 to gene A, and then gene C has a recombination frequency of like 0.3, well then you know the order of the genes are A, B's the closest, and then C's after that. You also have to look at recombinating with B, C, and B recombinating because it could be B's on the other side and C's on the other side. But I digress. I don't want to go too far down that rabbit hole, but you're getting the understanding that by understanding the recombination frequency between two genes, you can understand their proximity to each other on the chromosome. So in terms of degrees of linkage, unlinked will always show a random distribution of gametes. So you'll get that Mendelian output, right? Those Mendelian genotypic ratios when you do crosses with these. Expected recombination frequency is 0.5. You'll often get between 0.4 and 0.6. All right. Complete linkage, you get the expected recombination frequency is 0. In reality, you might get, depending on how many times you do the cross, you might actually get some amount of recombination there, but it's going to be very, very close to 0. And then partial linkage, like I said, close enough to cross over at times, but also may segregate independently at other times. Partial linkage is what we call it. Okay, so how can we experimentally determine linkage? So in order to do this, there's a few things we want to know ahead of time, and we want to make sure we're setting things up the right way. So first and foremost, this is always easiest when you have easily, readily observable known phenotypes. Okay, now with the advent of DNA sequencing and the ease with which we can do that now, it's not always required because we can follow along pretty easily with tail snips or depending on what organism you're working with, we can follow the genotype pretty readily doing that. But for this example, we're going to use some pretty readily observable phenotypes. We're going to look at tail length and coat color. So for example, here, the little a gives you the short tail, the recessive phenotype. The big B gives you the brown fur coat. And then it's the inverse over here, the white fur coat for the little b, and the dominant long tail, big A. So the other thing you want to do is make sure you know the genotypes of the parents. Okay, that is very, very important. And what you want to do is have contrasting alleles. This is on purpose that we have homozygous recessive, homozygous dominant, homozygous dominant, homozygous recessive. That is on purpose because that means when these two have offspring, we know what the genotype will be. It's going to be big A, big B, little b. Okay, so we know that that's the only possible combination we can get out of in the F1s. All right, so with the F1s, we get back this particular genotype, big A, big B, little b, heterozygous at both alleles. So what's the next step? Well, what we want to do in the next step then, okay, is we want to use what we call a homozygous recessive tester. This is an individual that is homozygous at both alleles. This is going to make it very, very easy for us to determine which phenotypes are parental and which genotypes are parental and which are the recombinants. Okay? So, how is this going to work? We're going to do a cross where we mate the F1 progeny that we know are heterozygous at both alleles. We're going to mate them into the homozygous tester that is recessive at both loci. And we're going to look at the offspring. Okay, so here's what we need to do. We first need to define what are the parental genotypes. Well, in this cross, the parental genotypes based on this are big A, little b, or little a, big B, little b, right there. Long tail white coat, short tail brown coat, right? Those are the parental gametes that come back out of this cross. The recombinants then would be the opposite of that. Double recessives, okay? And the combination of this, okay? I know you're thinking, but these are the genotypes we see in this particular parent. Yeah, I understand that. But what matters is that in this cross, we know what combinations have to be given, right, to come back out of this. So these are the parental genotypes. These are the recombinant genotypes. And you can see what they look like up here. So if we get results from this and we take a look and we only observe two different phenotypes here, we observe short tails brown fur. So the little a, big B, or we get long tails white fur, big A, little b. We know that there are no recombinants. No recombination events happened whatsoever. Okay? So that tells us that these genes are linked. Why? The recombination frequency is 0. There's nothing there, correct? Because in order to get these particular genotypes right here, based on this cross, the only way you get these is if a crossing over event happened during meiosis 1, during the formation of the gametes that came from these individuals. But since we don't see any recombinants, no recombination has occurred. No crossing over occurred. That tells us that they must be so physically close on the chromosome that the recombination frequency is 0, and we would say that these are completely linked. But let's look at what we could get out of some real data from this. Okay, let's say we do the cross, and the answer isn't perfect, right? If we get just as many recombinants as we get the parental genotype, we know they're completely unlinked, right? Or if we get no recombinants, we know that they are completely linked. But let's say we don't get either of these scenarios. Let's look at what real data may look like. So we do this cross, and we start looking at the phenotypes. Short brown, 48. We know that this is going to be the gamete. We know what this is going to look like, right? We know what that genotype has to be because we've controlled for that. We get long white, right? Long tail white coat, 42 of those. Well, we know that that must be another one of the parentals. Big A, little b, little b. We know because, again, we've controlled the genotypes of the individuals who are mating. But we also get some recombinants. We get 13 short white, 17 long brown. Now note, because these numbers aren't the same as these numbers, or even close, there's some linkage going on probably. So let's do the math and find out. So how do we do this? Well, we take the number of recombinants and we add them up. 13 plus 17. That gives us 30. We then take the number of parentals and add them up, right? We add that together. All right. And that would give us, I'll do quick math in my head, 90, I believe. Yep. And then we're going to take that and we're going to add to that the other combinations. 13 plus 17 is 30. That gives us a total of 120. So we end up with 30 recombinants over 120 total gamete pairs. Well, the recombination frequency in there then is 0.25. And that tells us that there's at least partial linkage going on. So a couple of things we know for sure just from this experiment. First off, we do know that these are in fact syntenic genes. They're located on the same chromosome because they are demonstrating at least partial linkage. And two, we know that they are reasonably close together on that chromosome. All right, we know that they are at least close enough to exhibit partial linkage, but not so close that they're completely linked to each other. it gets really neat, and I don't have time to go into it in this video, but you can do this with multiple genes. And this is how the earliest chromosome mapping was done. Before we knew anything about, in some cases, even before we knew about DNA and stuff like that, we just knew that we could do these genetic crosses. And sometimes things were completely linked, sometimes they weren't. And this is how we were mapping chromosomes before we really fully understood what chromosomes even were, which is wild. So I hope you get a better grasp of what linkage is, why it exists, and why it's really cool to geneticists, because it's a really old school way of understanding how chromosomal architecture is. So I really hope you learned a lot. I hope you were able to work through these problems. If you get stuck on anything, you can leave a comment. But I hope this helps, and I hope you enjoyed our conversation on linkage, and I'll talk to you soon.